Christina Warinner is professor of the social sciences in the Harvard Department of Anthropology, and she's also Sally Starling Seaver Associate Professor at the Radcliffe Institute. She and an international team of genetic scientists have made some startling discoveries about the ancient Maya city of Chichén Itza. Welcome, Christina.
Thank you. It's really nice to be here.
So you just published a story in Nature that seems to cast doubt on a lot of the very traditional stories about ritual sacrifices of virgins in the ancient city. Could you talk a bit about your findings?
Sure. So we started this project to investigate actually a very specific instance of ritual sacrifice at Chichen Itza. It was a chultun, which is a subterranean, usually water cistern, but sometimes they get repurposed for other uses. And this chultun was discovered back in the 1960s when they were first constructing an airport. And during the construction of the runway, they opened up this chultun and discovered that it was full of the remains of hundreds of children. And it remained kind of an enigma over the next many decades. There were few explanations for it. And it's located not that far away from the sacred cenote at Chichen Itza, which was very famously dredged and investigated in the early 20th century by an archaeologist at Harvard named E.H. Thompson. He's really the origin of some of the ideas that the sacrificial burials at Chichen Itza and other places were related to the sacrifice of women or sacrifice of virgins. And that became kind of popularized and repeated since the early 20th century. But there's been a really growing body of evidence that that isn't terribly accurate. And in fact, the kind of ritual calendar and the festivals and the ritual activities associated with the ancient Maya were very diverse and varied.
That's really interesting. Do you know if these were children who were buried there, how this myth of the virgins got started?
Yeah, so the kind of myth of the virgin starts back in around 1908 and really takes hold by about 1930 through a series of kind of popular books that are published. And the idea comes from reading colonial documents written by Spaniards talking about sacrifice activities in the 16th century. And so different scholars, but also travel log writers, look to this body of literature, this sort of colonial documentation, and projected that, hundreds of years into the past to Chichen Itza at its height around 800 to 1000 AD. So that was sort of the lens through which these remains, these archaeological remains, were being interpreted, although these archaeological remains predate these colonial documents by centuries.
But these bodies are of children, right?
Exactly. Well, Chichen Itza actually has many examples of human sacrifice at it in different contexts. So there's probably three quite well-known contexts. First of all, there's something called a tzompantli or a skull rack that is represented in downtown Chichen Itza. It's actually carved into stone. So there's a stone representation of these skull racks. Similar tzompantli are also seen in present-day Mexico City and Tenochtitlan. And actually, the physical remains of a real tzompantli were found in another part of the downtown area of Chichen Itza. So we know that these tzompantlis were real. They're skull racks, and they seem to have displayed the skulls of captives that were probably captured during warfare. So that's one context of ritual sacrifice. Another context of ritual sacrifice that is depicted is around the ball game and the sacrifice of ball game players, and that's also depicted in stonework. And then we also have then the sacred cenote, which was a very large natural sinkhole that was used over centuries by the classic Maya and the terminal classic Maya to make offerings of all kinds, ranging from offerings of jade and copal, precious stones, ceramics, all sorts of precious objects, and also of humans. And there's been some work done on that as well. And we know that there are both men and women present, and there are also people of different ages. So it isn't one consistent profile. What made the Chultun discovery so unique and so different is that the kind of nature of the sacrificial activity was so specific. It only contains children. They're approximately 3 to 6 years of age, so a very narrow age range. And One of the things we didn't know until we did genetic analysis is that all of them, or at least all the ones from which we were able to recover DNA, so we successfully recovered DNA from 64 of them, all 64 were boys. And then what was even more surprising is that many of them were closely related to each other, so brothers, cousins, and we even had two pairs of identical twins.
Wow, that is really fascinating. But how did you get involved in this investigation? What drew you to it?
So the way this project came about was a little bit of an indirect process. I've been working with a researcher named Rodrigo Barquera, he's a Mexican immunologist and archaeogeneticist who works at the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany. And also I'd worked with his boss, Johannes Krause, who's the director of the department there. We'd worked together for many years on different projects. And in fact, Johannes and I had worked about 4 or 5 years ago on a project to investigate the cause of the Cocolitzli epidemics that broke out in Mexico in the 16th century. And in fact, my dissertation research was on the Cocolitzli epidemic. So I've long been absolutely fascinated with the history of Mesoamerica and had a really strong background in colonial Mesoamerica in particular. And so this project actually began with a question about disease. What we were really interested in knowing, we had, through this first study, we worked on a cemetery that dated to the 1545 Cocolitzli epidemic, and we were able to show that it was associated with a type of Salmonella enterica infection. And we wanted to know if this left any footprints in the immunological variation, the genes that determine immunological resistance to disease in present day populations. And so this, in some ways, began as an attempt to get DNA from prior to the colonial period to make a comparison. But it came also with this incredibly interesting ritual context that was poorly understood. So through this project, we were able to address two questions simultaneously. One, looking at the genetic and immunological impact of colonial era epidemics, but also to try to better understand some of the ritual activities that took place at this really iconic site that remained enigmatic. And so Rodrigo had been working on some of these questions. As I mentioned, he's an immunologist, and he'd actually been previously responsible for doing tissue type matching for organ donation in Mexico in his previous career before he moved to Ancient DNA. And so he asked if I would join the project and work with him on this. And I said, yes, of course. This is an incredibly interesting and exciting project to work on. And it really allowed me to draw upon my training that I'd had in Mesoamerican archaeology and colonial history, also to try to help interpret the data, help kind of bring this story to life. And as the data rolled in, and as we started to really understand what we were looking at, it just became absolutely fascinating.
Wow. You're using ancient DNA, isotopes, and something called bioarchaeology. Could you describe these methods for a lay audience?
Sure. I mean, when we approach the archaeological record, we're really trying to reconstruct the lives of people who lived hundreds, if not thousands of years ago. And we want to know about how they lived, who they were related to, the kind of events that occurred in their life. But all these things are very hard to find out, as you might imagine. Many of the things that we do during our daily lives leave no traces.
Mhm.
So as archaeologists, we've had to become really creative at finding methods and tools that can try to get at aspects of these past lives. And so we can use bioarchaeology, which is really just the study of the physical human remains. And that's incredibly useful for identifying things like age at death, or also you can learn about pathologies that an individual might have. If, for example, they had a fracture that healed, or if they had trauma, or if they'd had an infectious disease that affects the skeleton. So just by looking at the bones themselves, you can infer quite a range of information about people in the past. And in this particular case, the most important information we were able to find was that the ages of these children, and also a kind of minimum count. It can be difficult when you have large burials with many individuals that are commingled to be able to determine exactly how many individuals are there, but we can use bioarchaeological techniques to try to do that. We also brought in stable isotopes, which is basically a way of measuring the bone chemistry of an individual that can give us clues as to the diets they consume during life. Different foods that people eat have a slightly different isotopic chemistry to them, and we can trace that People say you are what you eat, and that's true. We build our bodies out of the foods that we eat, and some of that isotopic variation that's present in the food then becomes kind of locked into your own bones, and we can measure that, and we can make inferences about the types of foods people ate in the past and similarities in diets. And so we were able to use that in this case to show that these relative pairs that we later identified had very similar diets. What was interesting is it wasn't that all the related children had the same diet, but each pair had a similar diet. Different pairs could have quite different diets, but the pairs always had very similar diets. And that was really a clue to us that they were raised in the same households. And then we were also then able to use ancient DNA for a variety of things. This allowed us to determine the chromosomal sex of the children, which is very hard to do using the skeletal remains. It is possible to estimate the sex of an individual when you have adult remains, and that's because the hormones that are produced during puberty will produce some characteristic changes in a person's bones. So for example, in females, the pelvis widens. In males, we get the expansion of muscle attachments on the bones. So there are some changes that occur as a direct result of hormones during puberty. But for young children, they haven't undergone these changes. And so the skeletons of young children are indistinguishable by sex. And so our really only way to know that is through genetic techniques. And then we were also then able to, through using ancient DNA, to identify relatives, to identify the twins. And we were able to look at the sort of immunological profile that they had encoded in their genes.
So how many of these techniques are new?
So some of them are quite established. So a lot of the bioarchaeological techniques have been developed over the last 60 to 70 years and refined over time, but many of those are quite established at this point. Stable isotope methods for paleodietary reconstruction are a little bit more recent. They really started in the 1970s, and by the 1990s and early 2000s really became quite mature as a discipline. And so that's another really robust tool in our toolkit. Ancient DNA really came online kind of last of those tools. So the earliest ancient DNA studies date to the early 1980s, but the method didn't really become powerful until around the 2000 teens. That's when there were a number of changes as a whole to the field of biology and a revolution in genomic sequencing that really makes it possible for the first time for us to really gain large amounts of powerful information from a genome. Prior to that, we weren't really doing genomics. We were doing genetics. We would look at tiny pieces of genes. We couldn't really look at the whole thing. We really are truly in the era now of genomics. We can really look at the entire genome. And that was a real game changer for us in ancient DNA. So really since like the 2000 teens, that's when ancient DNA first came online. But even within that, the ability to really robustly identify chromosomal sex, and especially to characterize these immunological regions, which are quite difficult to sequence in some ways, that is much more recent. More like in the last 5 years has that become something that we're really able to do reliably.
So you're really in the vanguard.
Yeah, we're right on the edge of the wave, which is a really exciting place to be, and also sometimes quite wild. And you're just trying to
It's
hold
quite
on.
wild how.
Well, it's interesting. This field, ancient biomolecular research in general, which includes ancient DNA, also ancient proteins, is something else I work on. It is changing so quickly. The techniques and technologies are advancing at such a pace that it's mind-blowing. I look back at my own dissertation that I finished in 2010, and none of the techniques I used are still used today. They've all been completely replaced, not once, but probably 4 or 5 times at this point. So it's incredibly exciting as a researcher. We're really in the golden age of ancient DNA right now. The things we can do are incredible. We're working on projects and answering questions today that 5 years ago I would have told you were impossible. But the pace of science is just moving so quickly. It is really, really fun. But it means we constantly have to work to stay on top of it because it does move so quickly.
That's fascinating. Is AI involved in this at all? People are always talking about AI.
No, right now we don't have AI involved in almost anything. We do use a lot of powerful computational tools. And one of the big transitions that's happened in the field is really this move from when I was working early on in my career. If you wanted to work in ancient DNA, you really needed intensive training in molecular biology and biochemistry because 90% of the work was at the lab bench trying to produce the data. And then the tiny amounts of data you produced, you could almost analyze on the back of a napkin. It was so little. And so the data analysis was really a minor component of it. That's completely changed. Today, the lab work is mostly standardized and can be done at large scale. We use a lot of robotics, but the data analysis is absolutely massive. When I was working on my PhD, if I worked really hard, I could generate about 100 sequences in a day, maybe in a few days. Today, our standard output for a single genetic sequencing run is 25 billion sequences. So that gives you a sense of just how much things have changed in the last 14 years.
Wow, that is incredible. What sort of challenges did the site itself represent?
I mean, the site is a World Heritage Site. It's extremely well known. Many people have visited it, which is absolutely incredible. And so I think it's exciting for people to then, they have a mental image of Chichén Itza when they read about the study, which is fantastic. They probably haven't visited the Chultun because it's kind of way off on the side and it's not really part of the public part of the archaeological site. And today it's not developed for tourism. But it was excavated in the 1960s and it's been in ENA collections ever since. And a lot of people have had questions about it, but didn't really have the tools or the ability to answer them. And that was what was so exciting, about 4 or 5 years ago when we started talking and negotiating and we're discussing how these new techniques were really opening up new possibilities. We began a discussion of, well, what if we actually tried to answer some of these really long-standing questions? And so we formed a team. That team grew over time. And we had also really fantastic relationships with the local communities who were very interested in this history. And we're really excited to be part of the study. So it was a really wonderful experience kind of putting this all together and now getting to share our findings with the world.
That's great. Could you talk a bit about your involvement with the local community?
Sure. So the project was really developed with local communities from the beginning. Many of the local communities had been working with some of our research partners that work in healthcare, participating in studies around, for example, diabetes and other long-term health concerns. And so when we first started the study, and we were particularly interested in looking at the immunological consequences of colonial era epidemics, they were also interested in participating in that and allowing us to compare their genetic variation to the people of ancient Chichén Itza. And in particular, what they were really interested in is whether or not they were closely related to those individuals. This is a real problem that we have in Mexico today, is that many, many tourists come from all across, not only Mexico, but internationally to see these amazing sites and to marvel at just how extraordinary they are. But oftentimes the local people in the villages, the Maya villages, they're not treated well. It's this real kind of disconnect between marveling at the ancient Maya, but not respecting the modern Maya. And so they were really interested in whether or not our techniques could also help demonstrate the connection that they have to these ancient peoples. And this was an important question too. There was a lot of population movement as a result of colonialism, and a lot of histories have become lost or muddled. So genetics offers an opportunity to try to clarify some of these historic processes.
What have you found?
So we found that actually they're very closely related to the people who lived at Chichén Itza. In fact, of all the populations that have been studied so far, the Tixcal-Cayub community, which is the one we partnered with, they're the closest genetically. So this does seem to be one region where there wasn't a lot of population movement, that the people that are living there today are from the same population that lived there 1,000 years ago.
That's fascinating. I hear that you also produced a comic book called Adventures in Archaeological Science. Could you tell me
Yeah.
about that?
Yeah, I'd love to. So this is a project I started in 2017, and initially it was just for an outreach event we were doing with the public about archaeology. And I was teaching a class at the time on scientific illustration. And so as a final project for my students, I had them design a page for a coloring book. I thought that would be fun and it would make them practice their drawing techniques, but also would be really fun for this outreach event and would allow us to also have activities for younger children. And they just did it a phenomenal job. So we ended up kind of putting it together. And the book itself features the different techniques we use in archaeological science with examples from all over the world, and introduces kids to what archaeological scientists do. And it's written in a kind of fun style to get kids to be interested and feel some of the excitement around archaeology. And so we did that for this event. It was hugely popular. We completely ran out of books like within an hour. And I thought, well, this is exciting. Maybe we could do a little bit more with this. So at the time I was working in Nepal, and I was part of this project where we were helping to distribute books to these libraries that were being built in the Upper Mustang, where they've traditionally had a problem of not having enough books for their schools. And they had a lot of donated books, but mostly from America. So they were almost entirely in English and Spanish, and they weren't in Nepali or Tibetan, which was what the children were actually speaking. And I thought, well, this is a real lost opportunity. We work in projects all over the world. What if we took our coloring book and we actually made it available in the languages of the people where we're working, and that could also be really helpful for us to engage better and explain what we're doing and get kids excited. And so one of the first translations we did was actually into Nepali and Tibetan, and we brought those on our field work, and they were hugely popular. And then from that point on, we had so many archaeologists volunteer to translate the coloring book into their own languages. We now have 64 languages. The coloring book's available in 64 languages, which is super exciting. And we've been working a lot more in Latin America now, and we have more than a dozen Latin American languages represented. We have 7 from South America, and we also have within Mesoamerica, we have Nahuatl, we have Yucatec Mayan, we have Mopan Mayan, we have Quechua, and we have Belizean Creole.
That is incredible. And in South
Yeah.
America, what do you have?
We have, I don't have my list in front of me. We have Aymara, we have Quechua, we have Yanesha, I have a whole list.
I
I
I
don't
think
remember them all off the top of my head, but yeah, we have 7 different indigenous languages. And of course, we also have Spanish, and we have Brazilian Portuguese as well. And we have French. So we have a good representation of languages spoken throughout the Americas, and we're really working on expanding our indigenous languages.
that is incredible. Do you have plans for other projects in Latin America beyond the books?
Yes, yeah. So we have more ancient DNA studies coming, and I'm really, really excited about this. My early career was in Mesoamerican archaeology. It's my first love. One of the problems that I faced early in my career was the techniques in ancient DNA weren't really advanced enough to be able to answer the kinds of questions I wanted to. In part because Mesoamerica is quite warm. It's located near the equator, and that's not great for DNA preservation. And it was just a technical limitation. Ancient DNA didn't preserve well enough for the techniques we were using at the time for me to really answer any of the questions that I was passionate about. So I ended up moving and starting to work in Europe and Asia and much colder places where DNA preserves really well. And I answered lots of questions about all sorts of different topics, ranging from the spread of dairying to population migration histories during the Silk Road. So I've worked on a lot of questions like that. But in the meantime, the techniques and the methods have improved so much that now these more tropical areas are really opening up to our techniques. And so about 2 or 3 years ago, I've kind of made a pivot to start to work again in Mesoamerica. A typical ancient DNA project takes 2 to 5 years to come to fruition. So this Chichén Itza study was the first one to reach a kind of finishing point for us, but we have many more ongoing, both in Mexico and in Belize. And I'm really, really excited. They're going to be focusing kind of on 2 periods. There's a series of studies focused on the classic period and trying to understand the migration histories and the relationship between the Maya and Teotihuacan. And also, we're going to be working on some colonial stories, trying to better understand the wide range of colonial diseases that were introduced.
Could you tell us a bit about your work in Belize?
Sure. So one of the things I've been working on is a project in the Belize Valley. This is a special project to me because it's the very first place I ever did archaeology. It's actually what changed the course of my career. When I was an undergraduate student, I actually studied microbiology and chemistry, and I thought that was going to be my career path. And I ended up going on an archaeological field school in Belize, mostly just out of interest. I had loved taking my anthropology and archaeology electives in college, and I had that opportunity to spend a summer excavating at a site or a series of sites in Belize, in the Belize Valley. And I did, and I absolutely fell in love, and I ended up changing my major and majored in anthropology, and it really changed the course of my career. Um, And I always love the sciences, so I've always tried to find ways of putting them back together. But I began, working. The first archaeological site I ever worked at was one called Baking Pot, which is in the Belize Valley. And a few years ago, I was approached by a team of people who have been working there and continuing to work there. Many people I remembered from, 20 some years ago. And they really wanted to know if ancient DNA could answer some questions that they had. And I said, yes, I think the techniques are there. So we wrote a grant application together, which was funded. And now we are in the process of generating around 300 to 400 ancient genomes for the classic Maya in the Belize Valley to better understand their kinship networks, their kind of social relationships. We have a really great study that we're working on that integrates ancient DNA, archaeology, paleodietary analysis using isotopes. It's a really ambitious and broad project. It's bringing together a lot of people to try to tell some lost stories of the classic Maya of Belize.
This is really interesting that you're excavating these lost stories. Do you have any plans to do the same thing in South America?
Not yet. I think it would be amazing to do so. And I've been talking to some people about some possibilities in the future. Right now, I have my hands pretty full, but perhaps in the future, that would be a possibility.
Well, we certainly hope so. Do you have anything to add that I haven't asked?
I think one thing I think is really exciting now is that over the past, you see, 5 to 10 years, there's been so many incredible advances in archaeology. So we've had this complete revolution in ancient DNA and paleogenomics, which is amazing for me. This is my field of research, and I find it really exciting. We've also had a complete revolution in remote sensing with LiDAR being able to basically see beneath the canopy, see beneath the rainforest. And this has completely upended our understanding of the population size and density of ancient people in Mesoamerica. And I find that extremely exciting. And in parallel, you have these tremendous advancements in epigraphy and being able to read the classic Maya texts. And so I think these kind of three things are starting to come together. And I think over the next 10 to 15 years, you're going to see some really, really exciting science and scholarship that's going to be able to unite the archaeology, the remote sensing, the ancient DNA, and the texts to tell very rich stories about the ancient Maya, about Mesoamerica that were just simply out of reach before. And I think it's just a really, really exciting time for this sort of interdisciplinary work. And I think we're right on the cusp of it.
Thank you so much. This is Faculty Voices. You've been listening to Christina Warinner. She's professor of the social sciences in the Harvard Department of Anthropology and also the Starling Seaver Associate Professor at the Radcliffe Institute. She and an international team of genetic scientists have made startling discoveries about the ancient Maya city of Chichén Itza. Thank you so much for being with us.
Thank you for having me.